Good morning, everyone. Today we're going to be talking about the nitrogen cycle. We'll be using the concepts we learned in biogeochemistry lecture last time and applying it to understanding the nitrogen cycle. So let's start talking about nitrogen and why it's such an important element for all living organisms. Nitrogen is essential for life. It's an important component of many structures of organisms, like muscles and tissues and organs. But it's also essential for the metabolism of organisms because it's found in amino acids that make up proteins, it's found in nucleic acids that make DNA, and very importantly for plants, it's found in rubisco. Rubisco is an enzyme which is basically responsible for carbon fixation. So rubisco is the enzyme that allows plants to turn energy from the sun into sugars and carbon, and that's basically what drives all of life. So rubisco is a very important enzyme and it's made up of a lot of nitrogen. So most organisms need more nitrogen than they can get. So organisms are always looking out to get nitrogen. In the same way that we as humans crave to eat meat and nitrogen-rich foods, plants and microbes are also hungry for nitrogen. In the last lecture, I talked to you about open cycles and closed cycles, and that I used the term element cycling for that. We also talked in the last lecture about understanding stocks, fluxes, inputs, and outputs to an ecosystem, and we called that mass balance. Finally, we talked about how organisms need different amounts of essential elements. We used the word stoichiometry or element ratios for that, and we talked about how this affects the rates of element cycling. Now, the nitrogen cycle is an example of a very open cycle. Nitrogen can occur in many different forms, and some of these forms are soluble, so they can dissolve in water. Nitrogen cycles through ecosystems, so it cycles from the air to the soil and out again to the air or to water bodies. But nitrogen also cycles within ecosystems, so it cycles within plants, between plants, from plants to animals to microbes and back to plants again. Because nitrogen is such a key element for all organisms, organisms tend to hold onto it when they have it. So it can be seen as a system of nested cycles. Nitrogen circulates within the organism, and it circulates between organisms within the ecosystem, and it circulates between ecosystems at a global scale. So whenever you're thinking about the nitrogen cycle, once you've defined the ecosystem that you're thinking about, then you need to think, what are the inputs to that ecosystem and what are the outputs to that ecosystem? So we're going to start by looking at the inputs. In our atmosphere, most of the atmosphere is actually composed of nitrogen. So nearly 80% of all the gas in the atmosphere is nitrogen. If you look at the little pie graph on the right-hand side, you'll see that after nitrogen comes oxygen, but there's an awful lot of nitrogen around in the world. The real issue with this is that the nitrogen gas in the atmosphere is not very useful. You can see in the little example in the bottom of how nitrogen gas is. It's got a triple bond between the atoms, which makes it very inert. It's very hard to break down and very hard to react with other elements. So in order for nitrogen to be usable, it needs to be fixed. The nitrogen gas has to be converted to ammonium or nitrates, because ammonium and nitrates can be assimilated by plants. So we use the word fixation to describe the process that happens when N2 gas is converted into a form that is usable by biological organisms. So therefore, in order for nitrogen to enter the terrestrial ecosystem, the nitrogen gas in the atmosphere has to be broken into reactive compounds. And breaking the bond requires a lot of energy. This occurs naturally in only two ways, and then humans have invented a third way to fix nitrogen. The first form of nitrogen fixation is called abiotic fixation. During lightning strikes, enough energy is supplied to break the bonds of the nitrogen molecule and to form nitrous oxide. You can see the chemical equation here. A molecule of nitrogen and a molecule of oxygen react with each other to create two molecules of nitrous oxide, and this requires energy. Some of this nitrous oxide combines with more oxygen to form nitrogen dioxide. And then that nitrogen dioxide dissolves in water to produce nitric and nitrous acids. Now those acids that are dissolved in water then come down in rain into the ecosystem and they enter the system. So that's an input of nitrates to the system caused by abiotic fixation. You can see the equation at the bottom. 2 nitrogen dioxide molecules plus water creates nitric acid. Now this form, abiotic fixation, accounts for about 10% of the nitrates entering to the within ecosystem cycle. And this form of abiotic fixation also actually occurs in internal combustion engines of cars because of the high temperatures that they achieve. So actually by driving our cars around, we are increasing the amount of available nitrogen in the ecosystem because we're inadvertently making a whole lot of nitric and nitrous acids that can then enter into ecosystems. So then the second way that nitrogen can enter the system is biological nitrogen fixation. So this is still nitrogen fixation, and it still requires a lot of energy, but it's done by bacteria. And only bacteria show nitrogen fixation. The equation below shows you that it's an energy-intensive process. You need nitrogen, you need hydrogen, and then you need 16 ATP to split just one nitrogen molecule. And you end up with ammonium. Not all bacteria can do this. Only certain bacteria can fix nitrogen. And they also, as I've mentioned, then need a good supply of energy. It's also interesting to note that because it requires ATP, its biological nitrogen fixation doesn't only require energy, it also requires that there's a source of phosphorus. So it's thought that nitrogen fixation is more likely and more common in soils that are rich in phosphorus. So this is an example of how cycles are linked. We will come across many such linkages in following lectures, where we're interested in one cycle, but then we see that the availability of elements from another cycle will affect the rates and the fluxes in one cycle. So here are some examples of the type of organisms that can fix nitrogen. There are 3 types of bacteria that can fix nitrogen. The first is rhizobia bacteria. You've probably already heard of these bacteria because they form a symbiosis with some plant species, so they're a really good example of a symbiotic relationship. These symbiotic associations are really easy to see if you dig up the roots. They look like little nodules on the root hairs. You can also smell them. It's very easy. We often, in the field, we're interested in our acacia trees and to see how old they are before they start to fix nitrogen and have these symbiotic associations. And you just dig up the seedlings. You can smell and you can see the nodules, and then you know where the nitrogen fixation is going on. Now, the reason it makes sense for rhizobia to form a symbiosis is because of how much energy it takes to fix the nitrogen. The plants supply the energy in the form of carbon, and the bacteria then make the nitrogen. This type of symbiotic association is only really found in some plant species, mostly legumes and peas. And that's why the peas and the legumes that you eat, and when we eat beans, there's a lot of nitrogen in those types of plant materials because those plants have access to more nitrogen than other plants do because of their relationship with the rhizobia. But there are 2 other types of bacteria that can fix nitrogen. You get free-living soil bacteria that can fix nitrogen, and also blue-green algal crusts. But even though these types of bacteria can fix nitrogen, they fix a lot less than the rhizobia do because they need to supply their own energy for the process. So you can see the numbers in the slide here. Rhizobia probably fix about 0.4 grams of nitrogen per meter squared per year, whereas the soil bacteria and the algal crusts fix less than 0.1 grams of nitrogen. So until about the 1930s, those really were the only 2 ways that nitrogen could enter the terrestrial ecosystem. But in the 1930s, 2 scientists called Haber and Bosch invented this process of splitting nitrogen from the atmosphere to form ammonium. And then when we discovered this process, which is called the Haber-Bosch ammonia process, we were able to produce large quantities of synthetic nitrogen fertilizers and also explosives and all other sorts of materials that require nitrogen. I'm going to talk about this more later in the lecture, but this was a transformative new technology. Nitrogen that had previously only entered ecosystems in small amounts through lightning and microbial activity could now be poured onto the landscape in huge quantities. But remember that this process is also very energy intensive. You can see in the diagram you have a heater there that is heating the system. So it depends on having cheap fossil fuels to make cheap fertilizers. So while this is a transformative process, it doesn't mean the nitrogen is free. Just like with biological nitrogen fixation, there's a cost to producing nitrogen fertilizer. The plant pays the cost in carbon, and we pay the cost in fossil fuels. So if we pause here and go back to thinking about the nitrogen cycle as a system of nested cycles, I've talked to you about the three main inputs of nitrogen to an ecosystem. In fact, the three only inputs: abiotic fixation, biological fixation, and artificial fixation through the Haber-Bosch process. So now I'm going to discuss how nitrogen cycles within organisms and within ecosystems once it has entered the ecosystem. Now, nitrogen is hard to get hold of, so plants and animals don't let go of the nitrogen that they have taken up easily. For example, plants, before dropping their leaves, will translocate as much of the nitrogen in the leaves that they can back into their stems and their phloem, so it can be used again to make the next leaf. The word translocation means the withdrawal of nutrients from senescing leaves and subsequent storage within the plant. I've provided a table here for you to look at so that you can see and get evidence for yourself of how plants translocate nutrients. So you need to be able to look at the numbers in this table and understand what they mean. So senescence is a word for aging. In the autumn, many leaves senesce and go from green to brown. And when the leaves go brown, that's because they have translocated all of the nitrogen-rich chlorophyll back into their structures. So have a look at this table. It's an example from a forest in North America. Before senescence, each leaf had about 7.1 grams of nitrogen per meter squared. After senescence, each leaf had about 3.3 grams of nitrogen per meter squared. And then there was some leaching of the nitrogen from the leaves during senescence. So if you minus 3.3 and 0.2 from 7.1, you get 3.6. So 3.6 of the 7.1 was taken back into the plant to be used again. So about half of the nitrogen that was in the leaf is still available to the plant to use again next year. Great, so we've discussed how the nitrogen is cycled within the plants. Now we're going to talk about how nitrogen is cycled within the ecosystem. There's a little diagram here of the within ecosystem cycle for nitrogen. Just to describe it in words, nitrogen in plant litter is decomposed by microbes, which can make it available for uptake by plants again. But in this process, nitrogen changes from an organic form into an inorganic form. In undergrad chemistry and first-year chemistry, you should be very clear about the difference between an organic and an inorganic molecule. And this translation from organic to inorganic is very key for understanding the nitrogen cycle. Plants are only really able to take up nitrogen when it is in an inorganic form. Another thing to think about is that this figure that I've given you is a systems diagram. All of the words are stocks. Microbes, litter, animals, plants, those are all stocks. And all of the arrows are fluxes. In the next few slides, I'm going to name these fluxes and talk about how the nitrogen transforms from an organic to an inorganic form and back again within the ecosystem. So there are three key fluxes that you need to understand in the within ecosystem cycle. And they all have long, complicated words, and we need to distinguish between these words. I remember when I was in second year, I really struggled to get on top of the difference between mineralization, nitrification, and immobilization. So I'm hoping that I'm going to do a better job with teaching you than I managed to understand in second year. So let's hope that by the end of the slide, everyone understands the difference between these three different processes or fluxes. The first one is called mineralization. And this is the process by which microbes decompose organic nitrogen into ammonium. That's why it's called mineralization, because you're taking an organic form of nitrogen and you're turning it back into a mineral. So this is the plant litter, or your rich organic nitrogen, and it gets transformed into ammonia and then quickly into ammonium. Now, ammonium is available to plants. So this flux turns unavailable organic nitrogen back into available inorganic nitrogen. However, once you've got ammonium in the system, then microbes can actually decompose that ammonium into nitrate, and they get a little bit of energy by doing that. So you have a second flux in the system, which is called nitrification. So here you take the ammonium that was created in the first step, and it gets turned into nitrate, which is also available to plants. So both of these processes create an inorganic form of nitrogen that's available to plants, but it's a two-step process. First, the plantlet is decomposed and mineralized into ammonium, and then it is nitrified into nitrates. So those are two processes and two fluxes that make nitrogen available to plants. The third flux that I want to talk to you about is called immobilization. Now that's the opposite. That's when the nitrate and ammonium that have been created are taken up by the soil biota. So basically, in order for these microbes and bacteria to grow, they also need nitrogen. So immobilization is when the soil biota use that nitrogen and take it up, so then it becomes unavailable to plants again because it's turned back into an organic form. Now you'll remember from your soils practicals that you actually calculated the mineralization rate of the nitrogen in the soil. So when you were doing that practical, what you were doing was you were calculating a key flux in the nitrogen cycle. Mineralization is a really important flux because it represents the rate at which nitrogen becomes available again to plants. So it is arguably way more important than measuring the total amount of the nitrogen in the soil, because from a plant's perspective, what's going to help a plant grow is having a readily available amount of nitrogen that they can take up. So often when you're trying to understand how ecosystems work, one of the key things you want to know is what's the mineralization rate? How quickly is that nitrogen cycling within the ecosystem? And one of the key things that controls how quickly the nitrogen is cycling within the ecosystem is the C to N ratio, the carbon to nitrogen ratio of the plant litter. So now we're going back to understanding our element ratios and our stoichiometry. If you remember from the previous lecture, we know that nitrogen occurs in characteristic ratios with carbon and phosphorus, and this is called stoichiometry. You also remember from the previous lecture that in plants, this ratio is not fixed. It depends on the type of plant and the type of plant part. So for example, woody tissue has a C to N ratio of 200 or more. So 200 carbons for each nitrogen. It can even go up to 1,000. Whereas leaves and fine roots have a C to N ratio of about 15 to 30. So that would be about 30 carbons for each nitrogen. However, in animals, bacteria, and phytoplankton, the C to N ratio is fixed, and it's at around 6 to 1, so 6 carbons for every nitrogen. So this imbalance in the element ratios between plants and the things that are eating the plants makes cycling of nitrogen from plants to animals and back to plants again very complicated. And it determines the rate of decay and the rate at which nitrogen will be available. Now I've mentioned all organisms want nitrogen, so microorganisms and plants are all competing for the available nitrogen. And the higher the C to N ratios, the higher the competition. So what you need to take away from this slide is to understand that depending on the C to N ratio of the material that's coming into the system, the longer it will take the microbes to digest it, and the slower it will take for the nitrogen in that plant tissue to cycle back into this ecosystem and be available for plants again. So here are some data to help you understand that concept of the C to N ratio and how it affects decomposition and how it affects nutrient cycling. These data are from a beautiful savanna reserve just north of Pretoria, quite close to where we went to the field, to the Tswayin crater. And in that savanna reserve, you have two types of trees with two types of leaves. You get the broadleaf trees that have big, thick nitrogen poor leaves, so they have a lot of carbon and a lot of structure and not that much nitrogen. And then you have the fine-leaved trees, like the acacias, that have thinner leaves that are more productive and which contain more nitrogen per gram of carbon. So what the researchers did is they gathered some litter from the broadleaf trees, some plant litter, some dead leaves, and they gathered some litter from the fine-leaf trees. And they put them on the ground and they saw how long it took for that litter to decompose. So every week they went back and weighed the litter to see how much of it was still there and how much of it had decomposed. And you can see in the graph, on the x-axis you've got time, and on the y-axis you've got the amount of plant litter remaining in the ecosystem. The broadleaf trees are the dotted line, and the broadleaf trees, the litter stayed in the ecosystem for a long time. It took a very long time for the litter to decompose. After 180 days, so after 6 months, there was still more than 60% of the litter waiting around in the ecosystem. It hadn't been decomposed. On the other hand, the fine-leafed litter, the litter that had a lot of nitrogen and a lower C/N ratio, decomposed very quickly. After 180 days, there was about 20% of the litter remaining. So this graph demonstrates how the stoichiometry, the element ratios in the plant material, affect the decomposition rate, and then that affects the rate at which the nitrogen goes back to the system. If you're a grass plant underneath an acacia tree, then you're going to get access to nitrogen more quickly because that acacia leaf is going to decompose, the nitrogen will be mineralized, and it will come back to the system to be available for new growth. If you're a plant underneath this is a Berkia africana tree, a broadleaf tree, then the nitrogen is still stuck in the leaf. It's not available for plants to grow, so that means that that system cycles more slowly and is less productive. Great, so we've spent quite a bit of time talking about the within organism and the within ecosystem cycle, because there's a lot of interesting biological activity going on within the ecosystem when it comes to nitrogen. But despite the fact that nitrogen is very much in demand in natural ecosystems, and it's taken up whenever it is available, there are still quite a lot of outputs in the nitrogen cycle. So there are a lot of pathways by which nitrogen can be lost in the ecosystem. And I'm going to discuss some of these outputs in the next few slides. So this slide gives you a little summary. It's a systems diagram showing you what all the different losses are and the outputs of nitrogen from an ecosystem. So if you see the big rectangular dashed line, that's what we would consider our ecosystem boundary. And inside that ecosystem, you've got plants and organisms using nitrogen and cycling nitrogen. And the fluxes are mineralization and nitrification, and plant uptake and immobilization, and all of those processes. But because ammonium and nitrate are actually quite reactive elements, you also have these fluxes out. So every time organic nitrogen is transformed into ammonium or nitrate, there's the potential flux out of the ecosystem. You can think of it as a leaky pipe with a small amount of nitrogen leaking out as it gets cycled around the ecosystem. And this is just unavoidable. There's no ways for nitrogen to be cycled more tightly in the ecosystems because you have these processes of volatilization and denitrification. It's basically, ammonium is an ion in solution in the soil, but ammonium can transform into a gas, which is called ammonia, and get lost to the atmosphere. So this is called volatilization, and then the ammonia gas goes up to the atmosphere and it's no longer available in the ecosystem. Nitrate is also an ion in solution in the soil, but nitrate can transform into a gas as well. It can transform into nitrous oxide or nitrogen dioxide or nitrogen gas, and it can get lost to the atmosphere. This process is called denitrification, and it depends a lot on the amount of waterlogging in the soil. You can have aerobic or anaerobic denitrification, and then the nitrogen will go up as a different form of gas, but either way it gets lost to the ecosystem. The third big loss in the ecosystem is via leaching. I've mentioned that ammonium and nitrate ions are both in the soil solution. They're very soluble. And you also get dissolved organic nitrogen that is dissolved and soluble and in the water. So anytime you get leaching of water from an ecosystem, where you get water moving into the groundwater and the rivers, that water can contain nitrates and ammonium. And so that's another loss to the system. And then there is one other way that nitrogen is lost in an ecosystem before it even enters the soil. When fires burn through vegetation, then a large proportion of the nitrogen in the plant biomass goes up as a gas. This is quite a small flux in most ecosystems, because in most ecosystems, fires only burn very rarely, not very often. But in African savannas, you can see in the map in this slide, many African savannas will burn every year or every 2 years. And so denitrification through felt fires is the main recycling process. It's called pyrodenitrification. So sometimes up to 60 to 90% of the nitrogen content of this tissue will go up into the atmosphere and be lost from the ecosystem. So you can see that with all of these losses, you've got volatilization, denitrification, leaching, and pyrodenitrification. It's a good thing that there are some inputs of nitrogen to the system, otherwise you'd end up draining the system of nitrogen. So now we've discussed all of the inputs and the outputs and all of the key fluxes and processes within the ecosystem and organism cycles. So this slide is a summary of the entire nitrogen cycle of a natural terrestrial nitrogen cycle. You can see that it's a systems diagram. We have stocks, and those are the colored items, and we have fluxes, and those are the arrows. Now I mentioned that it's very important if you have a lot of outputs to the system that you have an equal amount of inputs to the system, otherwise you're going to end up draining the system of nitrogen. So you can see the little numbers next to all of these fluxes, and theoretically in a natural, sustainable terrestrial ecosystem cycle, the inputs and the outputs should be approximately balanced. So have a look. You've got two inputs here. You've got the biological nitrogen fixation, which is about 0.8, and you've got N deposition. That's the abiotic nitrogen fixation, that's 0.5. So that's 1.3 grams of nitrogen coming into the system. Then what are the fluxes out? You've got pyrodenitrification, denitrification, and you've got leaching. So if you add all of those up, then you end up with about 1.3 grams of nitrogen per meter squared getting lost from the system. So in this natural terrestrial system, although you have a very open cycle with a lot of inputs and a lot of outputs, it's also in balance. So the total amount of nitrogen in the system is not changing too much. So it is true that in natural ecosystems are commonly limited by not having enough available nitrogen for the plants, and that's because of how open the nitrogen cycle is. It keeps leaking nitrogen. The other thing to note is how much more nitrogen is in the atmosphere than in the soil, and how much more is in the soil than in the plants, and how much more is in the organic form in the soil than the inorganic form. The amount of nitrates and ammonium is really tiny, and that's because that's the key resource. That's what's controlling the rate at which this nitrogen is cycling through the system. So what you need to take away from the slide, you need to look at all of these words, and you need to know what's pyrodenitrification, what's N deposition, what's lithophore. So you need to know the meaning of all of these terms. And you need to understand how the stocks and the fluxes link together, so how the nitrogen is cycling through this ecosystem. Right, so up till now, I've given a brief summary of the complex and important cycling of nitrogen within and through ecosystems. Now we're going to learn about how humans have manipulated this cycling and what that means for the global nitrogen cycle and for ecosystem health. As I've mentioned before, even though nitrogen is a very open cycle with lots of inputs or outputs, before human alteration, the N inputs and the N outputs were approximately balanced each year. About 100 teragrams of nitrogen came into the terrestrial system each year, and about 100 teragrams of nitrogen went out again through leaching and volatilization. So it was balanced. In the past century, however, human activities have doubled the amount of nitrogen fixed from the atmosphere. So they've doubled the amount of nitrogen coming into the atmosphere, coming into the ecosystem. Why have we done this? What is the point of adding all of this nitrogen to the ecosystems? Well, basically it's to make food and explosives and other things, but let's focus on food for now. When we grow plants to eat, we're actually taking nitrogen away from the ecosystem. So every time we harvest some mealies or some soybeans and take them away to eat, we are extracting nitrogen from the ecosystem. So we're creating another output, really. You can see this in the graph on the left-hand side. You can see how the amount of nitrogen in the soil decreases as the amount of time that a piece of land is being used for agriculture increases. So the only way to maintain agricultural production is to add fertilizer, because if you keep harvesting plants, then you're going to end up with less production and less good agriculture. Now, of course, we've been practicing agriculture for thousands of years, and we only learned how to create nitrogen fertilizer from the atmosphere, the Haber-Bosch process, less than 100 years ago. So how did agriculture survive before fertilizers? So early civilizations really had to rely on natural regeneration of nitrogen. People would plant their crops in places where annual floods would bring fresh sediments and new nitrogen into the system. In large parts of Africa, we practiced rotational agriculture. So we would cut down and burn and create nitrogen-rich sediment and then grow our plants for a few years and then leave the ecosystem to recover and leave the nitrogen to accumulate back into the system. People also practiced crop rotation, where if you plant crops that can fix nitrogen, like soybeans, then the next year you plant maize and other plants. So you can imagine that because we had to rely on natural processes to increase the amount of nitrogen in the system, it really constrained the amount of production and agricultural production that we could have. So you've probably heard of the Green Revolution, when we learned how to artificially fix nitrogen, the Haber-Bosch process. That initiated the beginning of the Green Revolution, which is by adding fertilizer to our agricultural fields, we could increase production dramatically. So we really dramatically increased the amount of food that we could produce on one patch of land by increasing our fertilization of nitrogen. And here's a graph to show you the impacts of the Green Revolution on the nitrogen cycle. This diagram's a little bit hard to understand, so it takes some time to think about it. What it's showing is the total amount of human-caused nitrogen inputs to the global nitrogen cycle over time, from 1960 to today. So we can divide the human inputs of nitrogen into 3 different types of inputs. Firstly, we've planted legume crops in all parts of the world where there didn't used to be legume crops. So every time we take a bit of grassland and turn it into a soybean field, we're increasing the amount of nitrogen fixation. It might be natural, biological nitrogen fixation, but it's still a human-created increase in the amount of nitrogen. And then, as I've mentioned, fossil fuel and burning of coal and driving around in cars, that increases the amount of abiotic nitrogen fixation. Remember, I told you that any process that creates a lot of heat interacts with the atmosphere and increases the amount of nitrates and that come into the ecosystem. And then finally, the large amount at the top, the amount that's been increasing over time, is nitrogen fertilizer through the Haber-Bosch process. So remember I told you that in the past there were about 100 kilograms per year of natural nitrogen fixed in the ecosystem every year. And you can see that we've more than doubled that. So now the total amount of nitrogen fixed through human processes every year is about 150 teragrams per year. So there's more than double what usually comes into the ecosystem is coming in of nitrogen every single year. So this one process, the Haber-Bosch process, truly has transformed the world. It's transformed agriculture, it's transformed warfare because now we can make cheap explosives, and it's also transformed ecology. The natural end cycle has doubled, and it's resulted in all sorts of consequences for the nitrogen cycle. But we also have to remember that this process depends on cheap fossil energy. We can only create nitrogen if we have cheap energy. So I mentioned that the Haber-Bosch process transformed our agricultural production, but also transformed our environment and our ecological systems. I just want to talk to you a little bit in this slide about what the increased nitrogen does once it comes into the ecosystem. Now you'll remember at the very beginning I told you how nitrogen is so rare and so hard to get hold of and how plants and organisms really try to get it. And nitrogen is a limiting nutrient in many ecosystems. So that means that when we start increasing nitrogen and adding nitrogen through deposition, we can increase productivity, we can increase the nutritional content of food, but we alter the species dominance. What you find is you find plants and organisms growing that are more competitive and that are more able to use the nitrogen. And the result of that is that you get a decrease in biodiversity. So when you get ecosystems that have a lot of nitrogen fertilization, sometimes from agricultural runoff or else from acid rain and deposition, what you find is you get less biodiversity because only the competitive plants that can really make use of that nitrogen end up dominating. And the other plants that were able to survive because they had clever, interesting ways of getting access to nitrogen or surviving at low nitrogen levels, they drop out of the system. So ecologically, what you find is an increase in productivity and a decrease in biodiversity when you add nitrogen to an ecosystem. But there's also consequences more generally for air pollution and for water pollution. You get eutrophication of water bodies, so you get all of that agricultural runoff from all of our croplands ends up in the water bodies and adding a lot of nutrients to the water bodies. You also get air pollution because once you've got lots of nitrogen in the system, then those leaky pipes that I mentioned get even leakier. So you get a lot of volatilization, you get a lot of denitrification, which means you get a lot of these nitrous oxide and nitrogen dioxide gases, and that means you get acid rain. And finally, many of these gases, particularly nitrous oxide, are greenhouse gases. They have a really high warming potential. So when you increase the amount of nitrous oxide in the atmosphere, you end up warming the planet. So it's really fascinating how many different consequences there are from just increasing the inputs of nitrogen to our ecosystems. This has been quite a long lecture. I've covered a lot of content. So I just want to talk through what I expect you to take away from this lecture. I went in quite a lot of detail just so that you could understand the process, but there are really only 3 key things you need to be able to achieve by the end of this lecture. The first thing you need to do is to be able to explain why nitrogen is often a limiting nutrient. So you have to be able to explain why nitrogen is important for different types of organisms and what uses nitrogen has biologically. And then you also need to be able to talk, like I mentioned, about the stoichiometry and how that creates constraints in how quickly nitrogen becomes available to organisms. You need to be able to list and you need to be able to describe all the inputs and the losses of nitrogen to an ecosystem. So remember the 3 inputs: abiotic, biotic, and artificial nitrogen fixation. And then the many losses. We had volatilization, we had denitrification, we had pyrodenitrification, and we had leaching. The third thing that you need to be able to do is to discuss the complexity of having different carbon to nitrogen ratios in plants and in other organisms. And then you need to be able to discuss why this makes within ecosystem nitrogen cycling complicated. And there you need to be able to discuss all the fluxes that I mentioned, the mineralization, the nitrification, the immobilization, and you have to be able to distinguish between organic nitrogen and inorganic nitrogen, and how this stoichiometry, the C-to-N ratios, limits or constrains the rate at which organic nitrogen becomes available and inorganic nitrogen to plants again. So that third point is really quite a detailed one. You can go back over the slides. You have to build up from understanding what the fluxes are and then what controls the fluxes. And what controls the fluxes is the stoichiometry. And then finally, you need to explain the consequences of artificial nitrogen fixation. So you need to understand that we've been pouring a lot more nitrogen in our ecosystems than have been in the past, and then talk through all the different consequences, from biodiversity consequences to pollution to greenhouse gases. So take your time to understand this. There's a quiz that you can go to check that you understand some of the key issues. And also, if you want some further reading, there's a really lovely summary that you'll just look at the next slide. There's a really nice, wonderful book chapter that you can read to understand these issues more clearly. Peter Vatusek and Pam Matson are two really influential and inspiring systems ecologists. And they wrote a book chapter in a book called The Princeton Guide to Ecology, which is available online and available in the Wits Library. And I just wanted to end with a quote from them. You can read through this quote. I'm not going to read it to you. But this book chapter really helps you understand the fascinating and interesting world of nutrient cycling and biogeochemistry and how the nitrogen cycle drives biology globally and also at ecosystem scale. So I hope you've enjoyed this lecture and you've managed to follow. I'll be checking on whether you guys are doing the quiz. And then the next lecture, we're going to be moving on to phosphorus and talking about another nutrient and how it cycles through ecosystems. Have a great day. Bye.